Well, I'd like to welcome everyone to this session. We certainly are interested in having new entrants into the generic drug space, and we are very welcoming to small businesses. And I certainly hope that this entire session will give you excellent information and the knowledge that you need to succeed in this area. Now, ensuring that patients have greater access to safe, effective, and affordable high-quality generic medicines is a public health priority for FDA. Creating more competition by increasing the choices of generic drugs helps reduce the cost of treatment and increases access to health care for more patients. Generic drugs continue to account for about 90% of all prescription drugs dispensed in the U.S., but they only account for about 18% of prescription drug spending. In the past decade, therefore, generic drugs have saved more than $2.2 trillion in healthcare costs to the public. Every year, we approve or tentatively approve hundreds of ANDAs, or abbreviated new drug applications, including many first generics and many complex generics. The complex generics are the harder to copy dosage forms, such as auto-injectors or inhalers and so forth. Because of the difficulty involved in developing these complex products, FDA has several enhanced efforts in place to maximize scientific and regulatory clarity for these products, to make sure applicants have the information they need to develop complete submissions that meet our standards, so that ultimately we can help improve patient access to these important treatments. And many of them have had less access in the past because of their complexity and difficulty in making reasonable copies. Because, of course, we want to ensure that the generic version delivers all the benefits of the original medicine. We know that increasing availability of generic drugs creates competition, which is why our continued efforts in the Drug Competition Action Plan, which we call DCAP, to remove existing barriers to generic drug development and market entry and to spur competition is such a priority. Under DCAP, the agency is working to continually improve the efficiency of generic drug development, assessment, and approval process, and reduce barriers for generics to enter the marketplace. This includes our activities to streamline the application process to reduce the number of assessment cycles needed to obtain approval, all without sacrificing the clinical and scientific rigor that underlies the generic drug program. In the past, there have been multiple cycles that products go through until ultimately receiving approval, sort of refining the application over time. But this is very time and effort intensive, and what we'd like to do is see people be able to get it right the first time and get a tentative approval or approval in a timely manner after submitting their application. The Generic Drug User Fee Amendments, or GDUFA, provides essential support for our generic drug program, enabling us to bring greater predictability, timeliness to the assessment of generic drug applications, and helping to accelerate competition, basically by giving us more resources so that we can help applicants. We can put out standards, we can put out guidance, we can answer questions in a timely manner for this very large program that's going on. GDUFA is not just about application review, but also includes a science and research program and numerous commitments that provide support to applicants before they submit their applications and help us to strengthen and diversify the pipeline of applicants, which I think is very germane to this group, including for complex generic drug products. In other words, our work to support the development of generic drugs under GDUFA begins long before the submission of applications, because often there's science that needs to be done to understand, for example, how to measure bioequivalence for certain non-oral dosage forms and so forth. And if we can solve these issues and put out guidance on what to do, then applicants can follow these instructions and they know they have a pathway. We do for research support the investigation of these generic drug development problems and challenges and help us establish and clarify the scientific framework for ANDA development, submission, and how we assess these ANDAs. Now, the COVID-19 pandemic has highlighted how important access to medicines is. Throughout the pandemic, we've maintained the FDA's standards of evaluating products based on the quality of the data and sound science, while continuing to meet our GDUFA commitments for reviewing applications in established timeframes. We also prioritized the approval of certain generics because there was increased demand during the COVID-19 public health emergency. And it really illustrated the role generics can play to step into the breach and provide these products, some of which are totally essential to health. And this is really part of our deep commitment to facilitating access to medical products to help address the critical needs of the public. Now, some people wonder about where a generic drug might be made, but whether drugs made in the U.S. or in different other countries, manufacturers must undergo the same rigorous application process, and the information is always fully reviewed by our highly trained scientific staff. We believe that maintaining oversight of manufacturing operations is critical to maintaining the quality of these drugs. And so we remain committed to using all the available tools to oversee the quality of FDA-regulated products and make sure that people have access. During the COVID-19 pandemic, we used some new tools, a novel of tools to look at manufacturing facilities. So we did remote assessments. We were all working online remotely much of the time. And so we had people, for example, wearing cameras going around looking at parts of factories and so forth. We got records. We did many different things to continue to have oversight of facilities and of quality, but not jeopardizing the supply of these drugs. And we are trying to resume inspections all around the world and have certainly domestically in many countries, but with the ebb and flow of waves of the pandemic, sometimes access to other countries is difficult. And this has posed some problems. As I said, we've tried to deal with, say, with remote inspections and other tools. And we are certainly prioritizing getting into inspections we need to do for pre-approvals for generic drugs. I'm really proud of the work FDA's done in response to the pandemic, and I'm also very grateful for the continuing efforts of the many individual companies in the generics industry who have worked to find solve the challenges and problems that arose, particularly shortage that arose during the pandemic. Now, as we move into the next phase of our response, we have the opportunity to examine more closely what was successful of all the things we had to change and to prepare for future challenges. So at today's forum, which is aptly titled The Current State of Generic Drugs, experts from across the generic drug program will provide insight and updates on emerging topics such as the pre-ANDA program, which is very helpful for developers, generic drug metrics, post-market safety, pre-approval inspections, and global generic drug affairs. So as I said at the beginning, I hope these sessions are informative and helpful to you as you continue your journey to help provide safe, effective, and affordable drugs to the American public. Thank you.
Thank you very much, Dr. Woodcock, for your insights and your encouragement and your continued support of our Generic Drugs Forum. I am very honored now to introduce our next two keynote speakers, Dr. Charlie Cho and Mike Kopcha. Dr. Cho serves as the Director of the Office of Generic Drugs, OGD, where she is the principal authority on all matters related to generic drug review and scientific advisor to the Commissioner and other agency officials. Previously, Dr. Cho served as Deputy Director of the Office of Study Integrity and Surveillance, OSIS, in CEDA's Office of Translational Sciences, OTS. With more than 18 years of experience in global drug development, Dr. Cho is an accomplished leader in both government and the private sector. Our next speaker after Dr. Cho will be Dr. Michael Kopcha, who is the Director of the FDA's Office of Pharmaceutical Quality, OPQ. This office has over 1,300 staff responsible for assuring the availability of quality medicines for the American public through assessment, inspection, surveillance, research, and policy. OPQ contributes to the assessment of nearly every type of human drug marketing application, including new drug applications, abbreviated new drug applications, and biologics license applications, including 351(k) applications, which is biosimilars. OPQ also performs the quality assessment of investigational new drug applications and establishes quality standards for over-the-counter drug products and facilities. Please, let us welcome our first keynote speaker, Dr. Cho.
Hello. On behalf of the entire Office of Generic Drugs, I would like to welcome everyone to the 2022 Generic Drugs Forum. I am Sally Choi, Director at the Office of Generic Drugs at US FDA. And once again, it is my pleasure that I share the keynote session with Dr. Mike Kopcha, the Director of the Office of Pharmaceutical Quality today. When I think about the current state of generic drugs, I think about the enormous efforts of both FDA and industry to ensure that patients who need safe and effective medicines have access to more affordable treatment options. Because of these efforts, we have made great strides in the arena of generic drug development to date. In 2021, the abbreviated new drug application ANDA program concluded another very productive year. Also, 2021 was another year our dedicated staff worked fully remotely, and you'll see that the pandemic challenge did not disrupt our work productivity. FDA approved hundreds of ANDAs, including some in record time. The program also set an all-time record with more than 10,000 supplement approvals, illustrating the generic drug program is healthy and the pipeline of generics is strong. Also, the pandemic has highlighted how important this work is now for more than ever. For example, since the beginning of the pandemic, we are close to taking the 1,500th COVID-related approval action and approving more than 90 original ANDAs for COVID-related treatments and therapies. For more specific and detailed achievements made by the Office of Generic Drugs and the Generic Drug Program, please visit the OGD's 2021 annual report website listed here. We also issued 149 product-specific guidances, PSGs, in 2021, 54 of which are for harder-to-develop complex products. To date, FDA has published nearly 2,000 PSGs, which can be found on FDA's website at Product-Specific Guidances for Generic Drug Development. As many of you are aware, PSGs reflect the agency's current thinking on the most appropriate methodology for developing drugs and generating evidence needed to support a specific generic drug's approval. These PSGs give applicants better opportunities to efficiently develop generic drug products and to prepare more complete and more accurate ANDA packages. FDA funded approximately $20 million in generic drug science and research programs. While we awarded science and research funding for new 6 contracts and 10 grants, we also continue to fund 10 ongoing grants and 20 ongoing contracts. Overall, OGD had 80 ongoing external research collaborations in 2021, with many projects that have been awarded in previous years continuing into 2021. All of these science and research efforts result in various scientific and programming innovations supporting our generic drug assessment work. Some of the examples are alternative bioequivalence approaches, including in vitro modeling and simulation, and alternate advanced B study designs that will aid time-saving, cost-effective way of preparing the successful ANDA submissions. Also, these efforts enabled us to approve those challenging complex generic products, such as long-acting injectables and generic inhalation products. In addition, scientific and regulatory collaborations are very important in this area, and we have hosted many public workshops, webinars, and forums such as this event, creating those collaboration spaces. In the past year, we had over 16,000 external stakeholders attending various events. The authorization of the generic drug user fee amendments, GDUFA 1 and 2, allowed the FDA to build a scientific and evidence-based foundation for the efficient development, assessment, and approval of generic drug products. You'll hear more about the success of GDUFA 2 as it comes to a close this fall, but at this time we have approved or tentatively approved more than 4,000 ANDAs, including more than 400 first generics and more than 500 complex generics. The expected reauthorization of a GDUFA 3 will enable FDA's generic drug program to continue its critical work to facilitate timely access to quality, affordable, safe, and effective generic medicines, with a focus on reducing the number of review cycles to advance earlier approvals, enhancing regulatory science, expediting complex generic drug development, and providing a solid financial foundation to support the generic drug program as it evolves. A central purpose of today's forum is to facilitate the critical collaboration between the FDA, the generic drug industry, and others interested in generic drug program development. It is our hope that providing regulatory clarity, offering practical advice and case studies, and discussing scientific issues related to generic drug development can result in more generic drug development and a clearer regulatory path to potential approval. GDUFA enables both industry and the FDA to carry out their important work in advancing generic drug development and approval. Ultimately, however, it's the American patient that benefits most from GDUFA-funded programs, since these activities contribute to timely approvals of critical and cost-saving generic drug products. Thank you to everyone who has worked hard to make this two-day event possible, and I hope the presentations are informative and useful for all the attendees.
Hello everyone, I'm Mike Kopcha. I'm the Director for the Office of Pharmaceutical Quality, or OPQ, which resides within the Center for Drug Evaluation and Research within the U.S. Food and Drug Administration. When I typically talk about quality, I always like to start off by noting that a quality product of any kind consistently meets the expectations of the users. Drugs are no different. However, in our case, the users of drugs are patients as well as consumers. Patients expect safe and effective medicine with every dose they take. Therefore, pharmaceutical quality then is assuring that every dose is safe and effective, free of contamination and defect. So in essence, it's what gives patients confidence in their next dose of medicine. Today I'll be discussing the present and future state of pharmaceutical quality, but more specifically, I'll focus on 3 topics. First, I'll talk about facilities and how we've handled facility assessment during the continuing COVID-19 public health emergency, including the impact to generic drugs by the alternative tools we've applied. Next, I'll talk about OPQ's continued innovation related to quality management maturity and ensuring pharmaceutical supply chains for U.S. patients and consumers. Finally, I'll share our work related to advanced manufacturing and future technologies. Certainly, the COVID-19 pandemic has changed the way most of us operate. We continue to face the constant flow of information, at times updated by the minute, which challenges our science and risk-based decision-making. However, one thing remains the same, and that is our mission to assure quality drugs are available to the American public. You can read about our response to COVID-19, along with other continued efforts and initiatives, in our 2021 OPQ Annual Report, which you can find online on OPQ's website. A key part of delivering on our mission is assuring the quality of the facilities making our medicines. Though many inspections were initially postponed, the FDA worked throughout the pandemic conducting mission-critical inspections. Still, many inspections couldn't be conducted after considering the safety of our inspectors as well as that of the staff at facilities. Though some inspections were interrupted, quality could not be interrupted. We needed to maintain the same quality standards, and to do so we employed a risk-based approach and alternative tools to inspection. The FDA's Resiliency Roadmap provided a detailed plan that has guided the agency in conducting necessary inspections, and we continue to follow this roadmap. I want to share how we handled facility assessments without inspections in many cases, and the tools we continue to use. Travel restrictions challenged our work, and facility assessments still needed to happen, so we had to be innovative. We relied heavily on alternative tools to inspection, such as requesting information in lieu of an inspection. We also relied on engagements with global regulators through mutual recognition agreements as well as confidentiality agreements. We also relied on remote interactive evaluations, or what we term RIEs. As explained in our guidance, RIEs use live stream video of operations as well as remote live interactions with operators to help assess a facility. Our alternative tools are working almost unbelievably to me, considering where we started. Alternative tools in part supported the approval of over 750 ANDAs and over 8,000 application supplements in 2021. Since the beginning of the pandemic, we've reduced the need for pre-approval inspections, or PAIs, by over 50%, which enabled the completion of over 250 application assessments. Of course, only a subset of applications, typically around 20%, have recommended pre-approval inspections in the first place. Though our alternative tools are working, they will never replace the gold standard of conducting an in-person facility inspection. Many do not know this, but OPQ leads pre-license inspections, or PLIs, for biotech products and participates in many pre-approval inspections for new and generic drug with the FDA's Office of Regulatory Affairs. In fact, last year, OPQ staff conducted over 40 pre-approval inspections and 21 mission-critical inspections over 13 states and 16 countries. It's easy to gloss over numbers on a slide, but keep in mind that in many cases, these inspections required our staff to endure long quarantines while isolated from their family and friends. The state of inspections is always changing based on new circumstances and information, but here's the latest information I can share. On February 7th, FDA resumed domestic surveillance inspections given the decline in COVID-19 cases, after developing a system to help us manage risks to FDA investigators and the regulated industry. FDA also continues to conduct mission-critical foreign and domestic inspections while continuing to leverage alternative tools. There is ongoing planning for additional foreign surveillance inspections, And of course, the global COVID situation is non-uniform and continues to change. We remain committed to the health and safety of our investigators. Let's consider that the point of an inspection is generally to confirm what is in applications and to assess compliance to current good manufacturing practices, or CGMPs. But compliance to CGMPs does not mean, for example, that a firm is investing in manufacturing improvements or deploying statistical control to prevent supply disruptions. Keep in mind that OPQ's mission is to assure quality drugs are available to the American public. Next, I'd like to talk about our program related to quality management maturity, or what we call QMM. QMM is the state achieved when manufacturers have consistent, reliable, and robust business processes to achieve quality objectives and promote continual improvement. It's important to understand that QMM, or quality management maturity, is not just one thing. It's an umbrella concept, and many elements fall under it, as is depicted in this slide. For example, quality metrics are a key aspect of a mature pharmaceutical quality system, data-driven approach to reduce quality issues and to drive continual improvement. However, QMM is about much more than any single one of these elements. One can think of QMM as part of an array of quality. FDA assesses drug product quality and applications, and we monitor pharmaceuticals in the U.S. market to ensure that each dose is safe and effective and free of contamination and defects, as I've defined earlier. This then gives patients confidence in every dose they take. Process quality is controlling manufacturing risks to provide a quality drug product from the raw materials to the packaged product. This then is what gives manufacturers confidence in every batch they release to the market. The FDA assesses process quality in applications, and we monitor and inspect facilities manufacturing for the U.S. market. Mature quality management uses a performance and patient focus to identify areas of improvement and implement those changes accordingly. This is then what gives manufacturers confidence that every batch they make will be acceptable to release to the market now or in years from now. Quality management then is an expectation in international guidelines, but the responsibility falls on the manufacturer. The quote that you see here on this slide was taken directly from the White House's 100-day report on supply chains. We have been challenged, OPQ have been challenged to develop a framework to measure and provide transparency into a facility's quality management maturity, or QMM. And in order to do that, we need ratings that recognize and reward manufacturers for having these mature quality systems. Systems that achieve sustainable compliance and focus on continual improvement. The bottom line is this: we need to incentivize improvements to the pharma manufacturing infrastructure that enhance the reliability of manufacturing and supply. Now, some have wondered if such a QMM program falls within the FDA's mission. Let me be clear that the FDA's mission is to protect and promote public health by helping to ensure that safe, effective, quality drugs are available. The key piece that I'm focusing on here is available to patients. Drugs are not available to patients if they are in shortage. So let me pause and emphasize one point that I made. You'd have seen many get wrong in the conversations and press articles about our QMM program. Quality management maturity ratings and quality metrics are not the same thing. They are related, but they are not the same thing. Quality metrics will be just one variable that informs quality management maturity ratings. As you saw in an earlier slide, QMM is the umbrella. Quality metrics is just one piece that resides under that large umbrella. So I want to be clear about that to avoid any further confusion going forward. The FDA has formed a cross-functional team to facilitate the development of a QMM program. Just a few weeks ago, we released a white paper that explains the importance of establishing a QMM program and the key challenges and elements needed to successfully implement that program. We'll continue to engage with stakeholders as we build this innovative QMM program. We will host 2 QMM workshops in May, so please register and join us if you can. We also hope to hold an advisory committee meeting on QMM at a later date. Stakeholder engagement has been a critical element in developing a CDER QMM program. The stakeholders impacted by a QMM rating program comprise the 6 Ps of the pharmaceutical supply chain, and they are pharmaceutical manufacturers, purchasers, payers, pharmacies, providers, and patients. Try to say that 10 times real fast. I may be biased, but it seems to me that nearly everyone in the pharma supply chain benefits from QMM ratings. Manufacturers with high QMM get recognition in the market. Purchasers and payers get more insight and confidence in the supply chain of the drugs or components they buy or reimburse. Patients, pharmacies, and healthcare professionals get medicine less at risk of shortage. And we at the FDA get to be better informed for resource allocation decisions, for example, inspection timing and frequency, and also around regulatory flexibility, for example, related to post-approval changes. Everyone comes away with more confidence than in the next dose of medicine. As emerging technologies are rapidly being developed, we at the FDA also need to be innovative to keep up with manufacturing technology. As many of you are likely aware, advanced manufacturing is a key component of the overall U.S. strategy to strengthen domestic drug manufacturing and also to increase the domestic supply of quality drug products. So let me be very clear on this point. Advanced manufacturing is not only for innovator products. Several of the companies engaged with CDER's Emerging Technology Program are or hope to be generic drug manufacturers. The FDA has long supported investments in advanced manufacturing to provide a safe and more secure drug supply. Of course, advanced manufacturing is not just one thing. It can be the use of innovative manufacturing technology or approach to improve drug quality and/or the reliability of the manufacturing supply chain. It could be the use of novel dosage forms to improve drug delivery and targeting. It can be the use of novel analytical tools to improve product characterization, quality testing, process monitoring, and/or control. Advanced manufacturing is not a futuristic approach out of Star Wars or Star Trek. Advanced manufacturing is now providing quality drugs to patients with cystic fibrosis, HIV, breast cancer, leukemia, and asthma. However, patients aren't the only ones who benefit from advanced manufacturing. It can enable better quality medicine by facilitating Six Sigma operation. It can improve the domestic economy by allowing for reshoring of drug manufacturing facilities. It can allow you to develop and produce drug products rapidly and prevent drug shortages. As the COVID-19 pandemic has certainly taught all of us, agility and flexibility are needed to maintain pharmaceutical quality in a public health emergency. We are currently leading our international regulatory counterparts in developing requirements for manufacturers exploring advanced manufacturing technologies. One example of our continued commitment to quality and innovation is the Emerging Technology Program, or ETP. That was established to offer pre-submission support to potential applicants. A major milestone for this program was the 150th FDA-sponsored Emerging Technology meeting. Of course, the technology cannot be emerging forever, and another recent significant milestone was the first graduation, if you will, from the ETP, and that was continuous direct compression, or CDC, not to be confused with the Center for Disease Control. CDC is a pharmaceutical manufacturing process that consists of dispensing, mixing, and compressing using equipment that is integrated, resulting in no breaks in the process. CDC can improve the assurance of product quality by minimizing human intervention and taking advantage of process analytical technology. Graduation means that CDC technology can now be fully handled by any assessment team within OPQ independent of the ETP. Due to the expanding workload of the ETP, we've begun implementing an ETP program enhancement, and we're going to call that ETP version 2.0 or ETP 2.0 to improve strategy, operation, and enhance communication with those adopting new technologies within the industry. We've also been working hard to develop a regulatory framework for advanced manufacturing. This effort, which we call the Framework for Advanced Manufacturing Evaluation, or FRAME, aims to provide clarity and reduce uncertainty for products manufactured with advanced technologies. The framework will need to address both current and potential future manufacturing innovation. Now, to identify these technologies, we funded the national academies to gather public input on emerging technologies and publish a consensus report. We need to know the technologies we might see over the next 5 to 10 years to help us identify gaps and pain points in the current regulatory framework. Thanks to the national academies and our emerging technology program, we know many of the technologies we will likely see. We've taken a phased approach to FRAME. First, we assessed existing guidance, regs, and statutes for areas of consideration. Next, we conducted in-depth impact analysis to make preliminary recommendations for the regulatory framework. Now we are in the phase of increasing our public outreach and soliciting public input to further inform our thinking and begin the implementation of components of this regulatory framework. We want to make sure that advanced manufacturing delivers on the promise of providing reliable, available, high-quality drugs for patients. In closing, although less publicized than the innovator drugs, we all know that generic drugs have played a critical role on our nation's response to COVID-19. Although we faced unprecedented challenges over the last 2 years and continue to do so, OPQ is now even more dedicated to the advancement of pharmaceutical quality. Simply put, patients deserve confidence in their next dose of medicine, and we remain committed to giving it to them. I'd like to thank each of you for the privilege of your time and hope that you enjoy the rest of the Generic Drug Forum.
Thank you for the great keynote presentations, setting the stage for the rest of the conference. My name is Ray Ford, working with Cedar SBIA, and I'll be your host and moderator for today's sessions. We'll now move into our first group of presentations. Our first presentation is on the use of knowledge-aided assessment and structured application, also known as KASA, in drug product assessment. Our presenter, Dr. Peter Capella, has over 15 years of pharmaceutical development experience from both the NDA and ANDA side, as well as nearly 15 years at the FDA supporting generic drug quality review across a wide range of therapeutic areas. He's been involved in important FDA initiatives like the PEPFAR program for prioritizing AIDS drugs for developing countries, as well as the current KASA initiative for structured drug product assessment. Our next presentation is on KASA in drug product manufacturing assessment. Our presenter is Dr. Rocky Shah, who works as an Associate Director for Regulatory Affairs in OPM-MA. She serves as the Principal Advisor to the Office and Deputy Office Directors and provides staff leadership and direction and assessment of manufacturing and facilities for ANDAs and NDAs, supplements including inspections to support application approval. Her prior roles in FDA include Branch Chief in OPMA, team leader in OGD, and senior research scientist in OTR. Following this presentation, our next presentation is on KASA and Biopharmaceutics assessment. Our presenter is Dr. Kimberly Rains, who serves as the branch chief responsible for the coordination and management of biopharmaceutics assessment conducted for the life cycle of FDA-approved PDUFA and GDUFA drug products. Dr. Rains develops CDER biopharmaceutic guidances, leads research projects within her division, and provides subject matter expertise to FDA policy initiatives. Our final presentation is on the integrated drug product assessment expectations. Our presenter, Dr. Mayra Pinheiro-Sanchez, possesses over 20 years of experience in CMC assessment of generic products in CDER and currently serves as Senior Pharmaceutical Quality Assessor in the Division of Immediate and Modified Release Products. 3. Office of Lifecycle Drug Products, Office of Pharmaceutical Quality. Mayra also serves as the ANDA Application Technical Lead in the Multidisciplinary Integrated Quality Assessment Team. Please join me in welcoming our first presenter, Dr. Capella.
Good morning, and thanks for that nice introduction. Today I'm speaking on the Knowledge Aid Assessment and Structured Application that FDA uses, known as KASA, and in particular, on my portion will be the Drug Product Assessment module. You'll be getting a lot of information in this and other talks, so I just wanted to at least have some concepts in your mind that you can kind of keep there during this presentation, and hopefully you'll see how KASA, as well as what I'm discussing today, will capture these concepts. Of course, it's FDA, so data is one of them. Structure, you'll definitely see that. Regulation and guidance, again, very familiar. Analytics, life cycle, and risk. And you can see we're kind of walking our way through this slide left to right, and there's a reason, because to me, all of these concepts are really pointing to one thing that is really what OPQ is all about, and that is a decision, right? A decision on the quality of the ANDA, for instance, that we have in front of us to assess. Keeping in that direction, assume that this is the mindset of a drug product quality assessor, and also assume that this is before KASA, so this is a number of years ago, for instance. You've got the ANDA itself, you've got a large document, a series of documents, hundreds, perhaps even thousands of pages of information to go through, and your primary tools to do that assessment are the regulations, of course, and the guidances. And the guidances over the years, develop and continually update. So assessors have always been at FDA looking at these three things very carefully and leading to a decision point. So again, that's kind of been a mantra throughout quality assessment at FDA. But now with KASA, as you see on this slide, there's a lot more detail, and it's in a very structured manner. So this is actually the index from the drug product assessment KASA that we use currently in Office of Lifecycle Drug Products. And I'll just briefly outline these topics for you. And then the rest of this talk will go into some actual screenshots of some of these modules, and I'll at least give you an update on exactly how we use these modules. Again, due to time constraints, I can't go through all of them and all in great detail, but you'll get a flavor of how we use this, this program. So we begin with an overview, which is basic information in the ANDA. It also includes a discipline summary, which is a compilation of our current thoughts on the particular quality of the ANDA application that we're looking at. The next few modules I'm going through are all related to the concept of critical quality attributes, and you'll see this as we walk down the list. The first one is, of course, the drug substance, which is probably one of the most important factors, right, in the drug product and its critical quality attributes. We also evaluate the excipients from a critical quality attribute status. and standpoint. And as you know, excipients are, very important in the development of good drug products. And of course, we have drug product critical quality attributes, which is consistently what you would think of as the specifications for the finished drug product, and those are assessed. Also, container closure, which again for solid oral dose is important. It may be even more critical as we go down the line in the future with things such as liquids, but for now it's certainly an important attribute. So if you combine all those pieces of the puzzle, so to speak, you're coming up to a drug product design, and KASA has a module specifically to look at the design of the drug product as well. Of course, as you noticed, for those of you familiar with ECTD, this looks very similar to what you'd see in an ECTD type of format, and that's probably not by accident. So the next module is stability protocol and study data, and specifically looking for trends and, if necessary, looking at failures, but trends in the stability primarily. There's also another module on comparability protocols, and on occasion we get these and we'll analyze them as needed. The one I left for last is the risk evaluation module, because in my mind, all these other modules kind of culminate and help support the risk evaluation. That's why in the previous slide, in the great arrow that was leading towards the decision point, I always think of risk as probably one of the key factors in that analysis. So I will definitely be going through this module towards the latter portions of this talk. Okay, let's get into the modules. Again, I know this looks very busy, but I will kind of break this down for you piece by piece, and you'll understand that as you see other modules, there's a lot of similarity in the structure. And believe me, this was also a learning curve for the assessors at the beginning, but now people are becoming more and more familiar with it and getting more comfortable with looking at it and using it for assessments. So this is the drug substance module. And again, the top portion of this slide, of this page, is what I consider navigation. So really simple information, ANDA ID, assessment status, and by that I mean, what point is the assessment? Is it an initial review? Is it with the primary assessor, secondary reviewer, or is it being finalized? So this really is just kind of a status of where things are at with the assessment. Now, since this is about the drug substance module, the next portion is really just an update on the drug substance drug master file, or DMF status. As many of you know, while drug product reviewers are looking at the ANDA itself, we have other review groups that are looking at the API drug master file. So we just want to keep in touch with them to make sure we understand at what point they are in their assessment. Now below you'll see what we list as ANDA specifications. So these are really the critical quality attributes for the drug substance. And this list really probably looks very similar to what you'd find in the application itself. And so it has release limits. It identifies the specification or critical quality attribute. Of course, the main difference, it has an evaluation, which is the assessor's viewpoint as to whether or not that attribute is dealt with adequately. And also to the far right of the slide, you'll see a number of boxes. This is our places where the assessor can include things such as reference links to the actual ANDA itself to point out certain data characteristics. Excuse me. Also, there's a box called SEC, which is where secondary can kind of comment back and forth with the primary as needed to get better clarity on the assessment. And the last box is related to information requests or any deficiencies that may need to be requested of the applicant throughout the process. So this format, again, while it looks very busy, is actually pretty well structured and can be looked at, as you've seen a few of these, very quickly. So we'll examine that on the very next slide. So this slide is the drug product module slide. As you recall, it handles the critical quality attributes of the drug product itself, or the finished drug product specifications. You notice the layout is very similar to what we just saw in the drug substance module. However, there are two acceptance criteria limits typically, and this is very common for a drug product, as you can recall, or you probably may understand from your previous experience, there may be a release limit and a stability or shelf life acceptance criteria. So we have the ability to capture those. Everything else is very similar to the other product. This, again, is mock data, so don't worry about this, just this test, just for information. But this is where the evaluation of the CQA would go from the assessor. And again, the ability to support that evaluation through reference links or secondary comments, and eventually, if needed, deficiencies or information requests. Okay, so hopefully by these two slides, you're kind of getting a sense of the structure of KASA as an assessor would see the exactly same thing. The next module I'm highlighting is called the Drug Product Design module. And this one is actually very close to what you'd see in the ANDA itself. For this case, this is the components and composition section. So you see, the identification of the ingredient, whether it's the API or an excipient, the actual grade. In certain cases, the particular grade of an excipient could have a great impact on its functionality within the drug product. The specific function of the excipient is listed, as well as a breakdown both by quantity and by percent of the components within the drug product. Again, very powerful, very good information to know about the design. This is a very simple IR model with a single layer. If those are more complex, like multiple layered dosage form, you would just see another instance of this table, and then you would see that further down. Also, there is an ability within KASA to copy and modify this information across strengths. As you all know, drug products come in many strengths, and this shows an ability of KASA to handle all of those variations. Okay, this module is the stability module, which looks at the protocol and design and, data trending. It's, again, very similar. The top is the navigation throughout the KASA. What you see in these blue lines here are actually where the drug product batch information would go. As you know, typically we look at 3 exhibit batches per strength for ANDAs. And so that information would go in this section, including the strength ID, the drug substance batch number and ID that was used to make the drug product batches, as well as the last third column, which is the URLs to the C of A's. So the certificate of analysis for the release of each drug product can be looked at through a simple click here. Below that, you'll see this is where we actually look at the stability data and have storage conditions listed. And I know this is very small to read, but the top one is typical ICH conditions at 2560. The lower one is accelerated, which would be 4075. And this is where we do our evaluation. So if we find, upon reviewing the data, some significant trending, for instance, we would capture that here and then make a note of it. And we actually capture it very specifically. So in other words, we would list the CQAs that are trending significantly. For instance, in this case, just as a mock example, I put assay and impurities. And then if you can read this, it's very difficult, but the assay is decreasing significantly, so that would be a flag to look at that more carefully. And the impurity, in this case, impurity degradant number 3 is increasing near its limit. So again, something we'd be on the lookout for from an assessment standpoint. So, and again, as you can imagine, this can go into multiple pages as the number of strengths increase, and if, again, unfortunately, if there's some stability issues. So, but again, it can be toggled from page to page very easily, and again, copied to other strengths as needed. This slide is where we talk about the risk evaluation module. This is really broken down to 2 parts, risk evaluation. That is, there's an initial risk assessment, which I'll go through, and then a revised or a finalized assessment. Now, risk assessment for drug product is split into 3 different groups. One is for physical characteristics or properties of the drug substance. The second is for chemical properties of the drug substance. And the third, finally, is a discussion or a viewpoint of dissolution and or release characteristics. In general, you could think that, and we at least believe that, release type does impact the risk overall for the drug product. So in this case, this example is a tablet ER or extended release. So its release may be different, its risk may be different than what you would see for an IR, which is immediate release, or a delayed release, etc. So these blanks are actually normally filled with information, and at this point, again, we don't have time to go through details, but what I'll let you know is that there's a series of questions and answers that come up, are created and developed by subject matter experts in the office, and these were created a while back, and they do support basically algorithms that are related to risk assessment. So depending on the answers to these questions and the assessor comments, they will build a initial overall risk assessment. So this is just the other two properties, the chemical and the dissolution, and you notice the format is virtually identical to the other physical properties. And as I mentioned earlier, the goal of this is to come up with this initial risk assessment score, so to speak, for each property. And again, apologies for the difficulty reading, but physical stability is considered a medium risk. Chemical is also medium risk for this product, and in vitro is a higher risk. Like I said, that could be depending on the answers to these questions, and could be the very nature of the drug product itself. Some are high risk, especially extended release. You can imagine the release profile is important and very critical. This slide has the revised or final risk assessment. And I say revised or final because it can change as there's a discussion between FDA and the applicant throughout the pre-marketing assessment. So if there's more information we'd like, we will request it from the applicant. So let's focus on only the physical stability due to the time constraints, but the other properties have the same exact format. So again, the structure is there. So again, you can't really read this, but this is the initial risk ranking, which is medium for this particular property of physical attributes. And this next two sections are called risk mitigation strategies. And these are basically ways that the applicant can demonstrate that they have this risk somewhat under control, either through by knowledge or measurement. For instance, like product design, you're looking at actual knowledge of the particular product design. So this may be studies, this may be a combination of literature, other aspects to help us get more confidence that the design is well understood by the applicant. Measurements focuses more on critical analytical methodology, for instance, and let's say if there is a critical quality attribute such as polymorphism or particle size of the drug substance, these would demonstrate that they have adequate methods to cover both of those concepts throughout the application. So this whole finalization of risk mitigation strategies and comes to a final risk update score if you have it. And in this case, for instance, it's because of the amount of mitigation that has been demonstrated in the application, it's reduced the risk from medium to low. So as you can imagine, the lower the risk, the more confidence that we have of the applicant's ability to handle that risk for the product going forward into the future. Okay, so that was really, again, a whirlwind tour of drug product assessment in KASA. Just, again, a few snapshots of the templates that we really use day by day. So let's look back at the drug product assessor's mindset, let's say, with KASA now. Again, the critical properties of having the guidances and regulations at our fingertips as we go through the NDA is important. But now a concept of life cycle has kind of come into the picture, and I know this is probably only the second time I've mentioned life cycle in this talk. However, if you can understand that now that we have these assessments in a very structured format within KASA, we have the ability to look across the life cycle of a drug product at how consistent we are as FDA in evaluating quality. And we still are coming to that all-important decision point, but now we are looking into a thing of a life cycle approach. And this is how it's done by analytics. And again, due to time constraints, I didn't really go through the analytics in each section, but a lot of these sections have the ability to do analytics across assessments. So we can do a systematic comparison with multiple assessments from multiple ANDAs, for instance, to give us a broad picture of quality across the entire life cycle of the product. And again, FDA, we consider the life cycle of the product from the outset of the RLD itself through its first generics and then finally to the last generic available. So we're looking for consistency across the board as FDA from a quality perspective. And so we see KASA as the tool that really gives us that ability now, whereas in the past, getting to that data was very, very time-consuming and painful and very challenging. So this really simplifies our viewpoint into life cycle and brings it into the forefront of our thoughts. In summary, let me give you 3 benefits of KASA for drug product assessment. It definitely provides a structured approach, as hopefully you've seen even this quick demonstration, and of looking at drug product assessments to fully support our decisions on quality. It also allows for collaborative viewpoint across the disciplines. We have 3 basic disciplines during quality assessment. The other 2 disciplines will be speaking, I think, right after this talk. But all 3 of us can look at each other's work pretty much in real time through KASA and to help for consistency in our decision making. And finally, it gives internal customers within FDA, and this would be someone like OGD or ODP post-marketing assessors, the really, for the first time really, the ability to review drug product life cycle information across the entire spectrum of the life cycle, using these analytic features to compare things like drug substance, drug product, critical quality attributes, stability information, data trending, and risk evaluation. So it's a very, very powerful tool that is just beginning to build. We're building more and more information into it as we speak. That's the end of my talk. I want to thank you for your attention, and we'll be willing to answer any questions at the end of the session. I do have one question for you, though, in the quiz. Challenge quiz question for KASA for drug product assessment is, which concept does KASA for drug product assessment support that was perhaps not previously in the mindset of assessors? So if you were watching the talk carefully, we had a mindset prior to KASA and after KASA. So the answers potentially are use of regulations and guidances, drug product life cycle from the RLD through all its generics, or FDA is an agency that is data-driven. So the best answer for this question is drug product life cycle from the RLD through its generics. So in other words, their ability to look at the data that we've always had, but we're never able to look at with any kind of efficiency. Now KASA has given us that efficiency and the ability to look at that. Okay, thanks again. Thank you very much.
Hello everyone, I am Rakhi Shah and I work at Office of Pharmaceutical Manufacturing Assessment as an Associate Director. Peter talked about KASA and how it is used to capture review in a structured format for drug product quality. I'm going to continue and talk about how we are using KASA to capture manufacturing assessment in a structured way and modernize our review practice. In this presentation, I'll cover use of KASA for generic drug application assessment, how we in OPMA conduct integrated manufacturing assessment. I will also cover how we do our initial risk assessment for facilities and manufacturing, as well as how we are capturing these assessments in KASA, including risk mitigation strategies. Finally, I would include some of the analytics we are using for our KASA for knowledge management purpose. This is our KASA homepage, and we are currently using only for solid generic application. For oral products, we perform our quality review in an integrated manner, and you learn what OLDP does in earlier talk. We in OPMA assess manufacturing and facilities, which is captured in this integrated manufacturing assessment module, whereas Office of New Drug Product assess biopharm information for the drug product, and you will hear from Kimberly in the next talk. KASA is an IT tool that allows us to capture critical assessment in a structured format, which then can be used to manage knowledge. For our integrated manufacturing assessment, this is the layout of our assessment template in KASA. It is arranged as shown in this slide. The first module is overview module, where we begin capturing some basic information for applications such as drug substance, drug product name, application number, strength, RAD and reference standard, type 2 DMFs used, and all the information about commercial facilities, including their name, address, FEI number, etc. Then we profile the facility based on the responsibilities listed on 356H form, which we cross-check with module 2 and 3 underneath drug substance and drug product facilities. Majority of the information here in this overview page is pulled by our automated IT system, and this is in turn pulled from 356H form. So our assessors do not have to duplicate in the review. This should tell you how important it is for you to fill out your 356H form correctly, and we have a guidance on them already. It is very, very important to include facilities information accurately. The next module is facilities assessment module. We begin to do all facilities risk assessment in this module. Based on the facilities from overview page and based on responsibilities, we profile, and based on the profile code given by our Office of Regulatory Affairs, and based on whether it gets coded as a drug substance or drug product or testing facilities, it gets populated in the sub-modules underneath facilities module. We evaluate all the commercial facilities used for drug substance, drug product manufacturing, testing, and primary packaging. In the next couple of slides, I'll talk more about how we perform our initial risk assessment as well as evaluate all these facilities in KASA. Next module is manufacturing risk assessment module. And in this module, we perform initial risk assessment for unit operation, which include all the factors, including drug substance, drug product, facilities, as well as unit operation specific, and how they impact product quality or CQA, or critical quality attribute. We are using quantitative framework, failure mode and effect analysis, or FMEA, for conducting initial risk assessment. Here we also conduct a risk assessment for microbial control, because that is evaluated by OPMA. Once we populate this module with all the unit operations used for drug product manufacturing, those unit operations then automatically populate into this next module, which is termed as unit operations. Here in this section, each drug product unit operation assessment is conducted, which includes equipment information, process parameter, in-process control, process development, and scale-up information. Next module is microbial assessment module, where the microbial assessment is conducted, and based on whether the risk scoring was low or medium for the solid orals, evaluation is conducted in abbreviated or full manner in this module. This other consideration module, which is our next module, is for conducting assessment on batch records, reconciliation, hold time, comparability protocol, process validation information if available, etc. Finally, we have our assessment summary, which is where the assessors write short summary on facilities and process. This also includes risk update table, which gets auto-filled. Initial risk scoring comes from this manufacturing risk assessment module, and the final risk scoring comes from other modules such as unit operation, facility, microbial, etc. This gets updated automatically as we are filling information in this module, and there is a risk update or justification that also gets auto-filled what we enter in other module. So the risk assessment, as I mentioned earlier, is based on FMEA framework, which is quantitative in our case. There are questions that are included in KASA that are specific for facilities compliance status, such as like current good manufacturing practices, data integrity, data concern. Also, it includes facility experience, etc. For manufacturing or unit operations, we have questions which are common for all unit operations that are pulled as a tier 1 questions. This will include drug load, scale up, facility experience, etc. And then we also have a series of questions that are unique to the unit operation. where assessors will enter the responses based on the information from the application. And then the built-in algorithms in the background will calculate the risk scoring, and this comes out as low, medium or high for facility as well as the unit operation. But here for unit operation, we are including facility information as well. So risk scoring could be termed as low, medium and high, and we have internal cutoff that we have identified. So if this actually scoring determines the extent of assessment that will be needed for unit operation or facility. So for example, if it is a low risk scoring, we would do abbreviated assessment. If the scoring comes out as high, then the assessor will do full kind of risk mitigation strategies and full assessment. If it is medium, depending on whether it was medium due to unit operation specific questions or facility specific question, we would incorporate our assessment in a similar fashion. Now going over how we determine initial facility risk assessment, we verify facility information with our internal database and see if it is named for the first time or if it is first application filed by the applicant. The dosage form could be new in certain cases, like example is facility had manufactured only oral tablet, but the new application might have listed them as a parenteral formulation manufacturer. In some cases, facility might have experience in similar dosage such as tablet, but in the application they may be using very novel manufacturing process or process control, for example laser drilling for use for the first time, and that is also accounted in our model. Our assessors also look into facilities compliance information. As I told you earlier, that is, it could have been a PAI inspection in previous inspection, but what kind of quality concerns were discovered in our prior inspections, any scale up or product specific concerns we might have recovered, received through field alert reports, FARs we call it, and or data integrity issues. This all goes into consideration into making decision whether we will recommend an inspection for the facility. It could be either pre-approval inspection, which is determined at the time of pre-approval if we feel that we need to see actual implementation of control strategy or actual things going on at the facility. We can also recommend post-approval inspection, which is actually to look at scale-up information. There could be some residual risk. We can approve application without inspection, but we still feel that we may need to go later to see its actual implementation when commercial batches are manufactured. So these two types of inspection are recommended by OPMA. There are some other types of inspection which are done at facilities, which include surveillance and for cause inspection, but they are recommended by sister offices such as Office of Quality Surveillance or Office of Manufacturing Quality. So I'm not going to go into those inspections. We have started using alternate tools currently, which could be used as in advance or in lieu of inspections, and this could include 704A4 pathway, where we can request facilities related documents, or RIE or remote interactive evaluations. We could also use foreign regulatory inspection under our mutual recognition agreement, MRA. In KASA template, the factors that I showed in earlier slide are all considered as drop-down unit options underneath all of the categories shown in this slide. So here facility, FEI, profile code, etc. gets pulled or auto-populated when we fill it in our overview page and facilities information page. And in this is an example of drug product facility assessment, where we look at prior facility experience and quality or data concerns, any other risk factors such as environmental could be one if we need to consider those. Our assessors select appropriate drop down from this, and the risk is calculated based on algorithm, and based on initial score the assessor can decide whether to recommend an inspection or not. There is no automated inspection recommendation based on this score. So this score just guides the assessors. Okay, and once assessor can recommend a PI or 704, they can request that, and then the request goes to ORA through our other, like they have to request through other database in Panorama, and then the inspection could occur. Evaluation is noted in the reviewer evaluation field, and every point you will see secondary assessor which will be able to comment on any concerns on primary assessor's evaluation. And this section mainly acts as communication mechanism within KASA for primary and secondary. For unit operation, as I mentioned earlier, properties of API, excipient which could impact manufacturability is considered. Also formulation factors are considered. We also look into whether process is reliable and process controls are assessed. With always we keep an end game in mind, whether the process is fit for purpose, control strategies are appropriate, and whether this whole process is scalable. In KASA, here is an example of lubrication or final blending stage. Here we link it with the FEI number, so we know which facility is doing this kind of unit operation. And we can now then start selecting from the drop-downs, which include type of blending. If the drop-down doesn't have the value, there is an option of other, and assessor can override that. We select equipment used, any additional comment, and then in process control, process parameter. This is a little bit manual right now, where the data is filled from the submission. It doesn't get auto-filled because we are not getting this data in structured format, but with our PQ-CNC effort, we are hoping that it will become easier in the future. Currently, assessors may want to fill in this data if needed, only if the unit operation is considered medium or high risk. In case of low risk, some of these fields get hidden, and assessor will not fill those information into the application, from the application. And they can focus on assessment, which could be minimum if the risk is considered low. This slide just is a continuation for the same unit operation, where we capture some other information, special consideration, then process development effort, any scale-up proposal, whether there is a scale-up on commercial manufacturing or there is no scale-up, and based on that, reviewer will assess the unit operation in this reviewer evaluation field, and finally they will pick the score or assessment, whether now the risk, which was probably medium or high, could have been lower based on all these control strategies and consideration of process development and formulation development. So this is done manually right now currently, and here you can see there is a deficiency field. If we need to send information request or deficiency, we open this module and write our deficiency, and we get auto-pulled at the end of our review, and then we can send it to our BPM to send it out to the facility, to the applicant. So here is a schematic of how we can use data analytics from KASA. Here is one example of, say, CQA dissolution, and different unit operations, like for example wet granulation or compression, what kind of process factors applicant has used, what kind of facility factors applicant has used to, say, mitigate the risk for this CQA with this unit operation, and that gets captured in a structured fashion. We can pull this information out, and we can look at this when we are, say, assessing a supplement for this application. So this really helps us in forming this great knowledge database, which can be used to assess our future applications. We are also using KASA analytics for facilities, and here it shows that with additional information for sites, for example, sites capability for manufacturing various dosage forms, compliance history, control strategy, unit operation used at that facility. We have that database now through KASA as well as some other databases available in FDA. And now we can compare any pending facility assessment or if the same facility is listed in that application, and we can readily see the facility's experience, and we can determine that proposed site has demonstrated capability and say process control strategy is in alignment with the prior information. We will deem it as a low risk, whereas if it is not the case, we may deem it as more scrutiny needed. It may not be necessarily that we would apply for pre-approval inspection, but we just need to look into that facility information more thoroughly. So with this information, we truly believe that KASA improves overall efficiency and helps making regulatory decision by improving the facilities as well as the manufacturing knowledge management. We had data, we have all this data, but it's all hidden in our Word template, where it takes forever to skim through that data when the supplement comes in, for example. So with this, we hope that we would have a better use of the data that we are capturing in FDA. So with the end, we believe that finally, to conclude my presentation, how I hope I gave you how we are modernizing our manufacturing assessment using IT advancements, and how the structured data and knowledge management can ensure consistency and objectivity in our decision making, which then leads to drugs with reliable quality throughout the product life cycle. With this, I really wanted to thank you for your attention and also thank my OPMA as well as OPQ KASA team, including IT team, because without their help, this dream couldn't have been a reality. So I thank you all. Here is a challenge question for you. True or false? KASA improves overall efficiency and helps making regulatory decision by improving the manufacturing and facilities knowledge management. The answer is true. Thank you all.
Thank you for the invitation and opportunity to present at this year's SBIA Generic Drug Annual Forum. My name is Kimberly Rains, and today I will discuss the use of Knowledge-Aided Assessment and Structured Application, KASA, in biopharmaceutics assessment. The following objectives will be covered in this talk. Describing how KASA is used in the assessment of biopharmaceutics information submitted in abbreviated new drug applications, demonstrating the biopharmaceutics risk assessment decision-making process within KASA, and providing the advantages of enhanced risk-based communication towards patient-centric quality standards in the KASA interface. To begin the discussion, I will start with the KASA pillar and structure, supporting our initial goals that KASA for ANDA solid oral dosage form provides transparency and consistency across applications for in vitro dissolution specification setting based on product understanding and biopharmaceutics risks to the drug product. Now that you have the overview, I would like to move our focus to more in-depth understanding of the KASA interface. Here is a snapshot of the biopharmaceutics KASA. It is structured with 6 assessment modules. There's the incorporation of comparative in vitro release profiles and PBPK IVIVC modeling reports. It's a question-based biopharmaceutics risk assessment ranking and database lifecycle management are just some of the key features. Now we will dive a little deeper into the ANDA KASA for Biopharm structure. Here I have displayed the 6 modules in the Biopharm KASA. I won't go over all of them in great detail, but I will illustrate the organization and the centralized focus on risk. KASA begins with an overview module. This module is pre-populated with application information. The next module, Reference Biopharm Properties, contains relevant label information. For example, information related to clinical pharmacology properties. The next module, Biopharm Considerations, provides more information on biopharmaceutic properties such as solubility and BCS classification. The heart of the assessment is in the risk evaluation. Here, the biopharmaceutics risk assessment focuses on the evaluation of potential in vivo performance impact due to quality design and controls of the drug product. Following the risk assessment, we have the in vitro drug release specifications. This is where dissolution method and acceptance criteria are captured in a structured format. The Biopharm KASA is concluded with the assessment summary, which provides a concise summary and a tool for communication of life cycle management information. We'll now focus more into the heart of the KASA for biopharm, the risk evaluation. The developed risk evaluation is based on principles described in ICH Q6A. These principles related to in vitro dissolution are not new, and we carry this through in the overall assessment of our ANDA KASA. We start with a calculated initial risk. This is a quantitative initial risk that is established at the initiation of our assessment. When this initial risk is known, we move through the assessment looking at various mitigation strategies that are in place to control any inherent risk in the drug product. When mitigation strategies are identified, we can then determine a final risk score. This final risk score would allow us to understand what risk remains in the drug product, even when all of the controls are adequately mitigated. The acceptable risk levels from a biopharmaceutics perspective are very low, low, and medium. As we continue this discussion, I'll go into some of these categories. I will now begin by walking through one of our question-based decision trees for initial risk assessment for biopharmaceutics. This decision tree is referenced for immediate release solid oral dosage forms. The initial question is related to solubility. Is drug substance's solubility high per BCS guidance or ICH M9? If the answer is yes, it is highly soluble, we next move to a question related to dissolution. Is the in vitro dissolution rapid in 500 mL of 0.1 normal HCl? This method is akin to the method in the FDA guidance for highly soluble drug substances. If rapid dissolution is achieved, we would consider that product as a very low risk. However, if they're unable to achieve rapid dissolution, this would be characterized as a low risk. I'll next move on to instances where solubility is not high or demonstrated to be low. That following question is pertaining to permeability. Is the drug substance permeability high? If permeability is high and in vitro dissolution of the highest strength shows rapid dissolution in a medium in the pH range of 4.5 to 6.8, those would be considered low risk because these weak acids, when they're dissolved in the stomach, should perform like a highly soluble drug substance because of their pH-dependent solubility. However, if it is not rapidly dissolved in the pH range of 4.5 to 6.8, or permeability is shown to be low, we would next look at critical bioavailability attributes. Critical bioavailability attributes, or CBAs, are defined as formulation or process variables that expect to critically impact the bioavailability of a drug product. If CBAs can be clearly identified, detected, and controlled, those products would be deemed an initial medium risk. However, if you are unable to identify, detect, or control any of these CBAs, this would place the product into a high-risk category. We have further delineated the high-risk category into two sections based on if it is possible to develop an IVIVC, in vitro in vivo correlation or relationship, or if there is any additional in vitro or in vivo data to support this final drug product. Based off the decision tree, you can see that we had 5 different biopharmaceutics risk levels for the initial biopharmaceutics risk categories. I'll next walk through some examples of the biopharmaceutics risk mitigation approaches for each of these levels. We'll start with very low. If the initial risk is very low, a standardized dissolution test with minimal additional information is needed to support this case. When the initial risk is low, adequate method development to justify dissolution method and acceptance criteria are needed. When the initial risk is medium, the in vitro approach is used to mitigate the biopharmaceutics risk. Dissolution tests should target to detect meaningful changes in identified critical bioavailability attributes to provide insight into the in vivo performance. When the initial risk level is deemed as high, an example of mitigation strategy could be the development of an IVIVR in vitro in vivo relationship to support patient-centric dissolution. This could be based on additional in vitro or in vivo data or physiologically based biopharmaceutics modeling. When the initial risk is very high, the mitigation strategy that is preferred is in vivo studies used to develop an IVIVC in vitro in vivo correlation to support patient-centric dissolution tests. The use of KASA provides added benefit to the biopharmaceutics assessment, in which regulatory flexibility based on risk rather than manufacturing capability can be employed. I've provided a couple of examples here to illustrate this point. The first is a comment that could be received from the agency. Provide justification to support a wider dissolution specification for an immediate release drug product containing highly soluble drug substance. Based on a risk-based paradigm, probable responses could be a scientifically sound explanation for root cause of slower dissolution, such as borderline BCS solubility/sink conditions, tablet surface properties, wetting issues, cone formation due to excipients or granules, or drug-excipient interactions. Another example of a comment from the agency could be, provide evidence or information to demonstrate no impact on in vivo bioavailability, bioequivalence, and/or safety and efficacy. Acceptable responses could be provided in the form of additional in vivo bioavailability, bioequivalence study, in vitro in vivo relationship, physiologically based pharmacokinetic modeling and simulation sensitivity analysis, exposure response relationships, for example, Cmax is demonstrated not to be critical for efficacy, or information demonstrating that there is no change in Cmax or Cmin for chronic use. As we've now walked through the risk evaluation module of the Biopharm KASA, I'll now move on to the in vitro drug release specification module. As I stated, this module contains the in vitro dissolution test method and acceptance criteria in a structured format. As you can see, we can capture relative strength, apparatus, rotational speed in unit, temperature, volume, and acceptance criteria in a structured manner. We are also able to compare changes within the review cycle. As you can see from here, we have iteration 1, which is the original review, in comparison to the changes that took place in iteration 2 of the IR response. As you noted, the acceptance criteria has been updated, as well as the media. Not only does KASA allow us to have the structured format for the dissolution acceptance criteria and method, we also have the ability to incorporate dissolution profiles and perform comparative analyses and incorporate it into our review. Within the Biopharm KASA, we have key considerations for selecting an appropriate dissolution method and acceptance criteria. One being the therapeutic use of the drug product, consideration if it is a narrow therapeutic index drug product or a high-risk drug substance, evaluating the onset of action, as well as evaluating the efficacy and safety profiles of the drug product. Also taken into consideration is the drug release and absorption time frame, where we're looking at if the product is an immediate release, extended release, delayed release, or a combination of such. We look at the rate limiting step for absorption, which could be characterized by the Biopharmaceutics classification system or any complex modified release formulation design. Also evaluated is the predictive performance of the dissolution method, as well as the capability of the dissolution specification to reject batches with unsatisfactory product quality. You may question or wonder why dissolution is evaluated to such a great extent. And it is because dissolution is important to informed quality and life cycle management. In the cartoon shown here, you can see all of the different areas which are informed and dependent upon dissolution testing. From pre-formulation to post-approval changes to biowaivers and in vitro in vivo correlations are all predicated and dependent upon proper dissolution studies. In the division of biopharmaceutics, we look at dissolution beyond its role as a standard quality control test. We look at the potential that it has as a patient-centric product quality specification, where we'll be able to use information from the in vitro dissolution tests to inform us on the bioavailability, bioequivalence, as well as serve as transitional modeling strategy and support to understand in vivo performance related to safety, efficacy, and systemic exposure. We see KASA for biopharmaceutics assessment as a great opportunity to continue patient-centric quality standards implementation consistently across our ANDA submissions. With the incorporation of the risk evaluation in the biopharmaceutics assessment, we're able to leverage previous and new guidances with regards to developing a more robust dissolution acceptance criteria and method specific for each drug product. Guidance revolving around IVIVC development, which was released in 1997, for products that demonstrate a very high risk initial biopharmaceutics risk. We also have guidance for the very low risk in the dissolution testing and acceptance criteria for immediate release solid or dosage form drug products containing high solubility drug substances. Additionally, for those products that are considered high risk, we recently published the draft guidance in 2020, the use of physiologically based pharmacokinetic analyses, biopharmaceutics applications for oral drug product development, manufacturing changes, and controls. We hope that a future KASA will continue allow us to support the product patient-centric quality standards. To conclude this presentation, I've prepared one challenge question. The question is, what does KASA provide for the ANDA biopharmaceutics assessment? structured knowledge to facilitate biopharmaceutics risk assessment, scientifically sound biopharmaceutics risk assessment focusing on clinical performance, enhanced risk communication towards patient-centric quality standards, improved assessment efficiency and consistency, or all of the above. The correct answer to the challenge question is all of the above. I would like to make the following acknowledgments. Development and implementation of KASA is a group effort. I would like to highlight the contributions provided by the following members: colleagues and staff in the Office of New Drug Products, the Division of Biopharmaceutics KASA Working Group members and user acceptance testers, and finally, the Office of Pharmaceutical Quality KASA Steering Committee. Thank you for your time and attention. It's been an honor to present in this meeting.
Thank you for the introduction. The topic of my presentation is integrated quality assessment, and I am going to speak from the perspective of drug product. The learning objectives include to describe the functions of the drug product discipline within the IQA and outline critical aspects of ANDA assessment, to identify common deficiencies and steps to avoid them, and to evaluate a case study to emphasize the importance of product knowledge and understanding. And I will do this within the scope of solid oral dosage forms. So our main goal is to ensure patient access to high-quality medicines. So how can we attain these high-quality drugs? First, the pharmaceutical industry should strive to manufacture high-quality drugs that meet established quality standards. These products should be manufactured based on sound science and taking a risk-based approach to product development in order to generate product and process understanding. And these will lead to the establishment of an adequate control strategy to ensure a product of consistent quality is manufactured. This will also provide a means for continuous improvement to strengthen the knowledge foundation. Second, the agency needs to provide the necessary regulatory oversight to ensure the availability of high-quality drugs to patients, and this will be the focus of my talk today. In OPQ, we employ science and risk-based policies and standards related to drug product quality through a multidisciplinary integrated quality assessment, also known as IQA. The KASA presentations provided details on the work of each discipline, so I will only discuss briefly the disciplines involved, and these include drug product, who is responsible for the assessment of drug substance controls, drug product design and control, container closure and stability. Manufacturing is responsible for the integrated assessment of process and facilities, and also the assessment of microbiology for sterile drug products. And Biopharmaceutics performs the assessment of product performance characteristics as applicable. So the team is responsible for generating a document that represents the state of quality of the subject drug product to ensure a safe and effective medication for the intended population. And this process requires that we excel in communication and collaboration, not only across disciplines directly involved within the IQA, but we also collaborate with our colleagues in lifecycle API, labeling, and others for our comprehensive evaluation of the ANDA submission. This process also supports our One Quality voice and our commitment to quality. And the remainder of my talk will focus on the aspects related to the drug product discipline. So let's start with the drug substance. As mentioned, the drug product assessor will evaluate drug substance control, and the drug substance information is generally generally referenced to DMF, which is assessed by the Division of Life Cycle API. And the drug product discipline evaluates the critical aspects of the drug substance in collaboration with the DMF assessment team. The drug substance information in the ANDA submission should focus on those attributes that can have an impact on the quality, safety, and manufacturability of the finished drug product. For compendial drug substances, controls established should, as a minimum, comply with compendial requirements. And based on product development studies, consultation with the DMF holder, and using a risk-based approach, additional attributes should be controlled. One of the most critical aspects is the knowledge and understanding of the impurity profile. Impurity limits should comply with ICHQ3A. And for genotoxic impurities, limits as referenced in ICHM7 should be considered. More recently, the presence of potentially genotoxic ASAT impurities in at-risk ARBs or angiotensin receptor blockers has been identified, so these should be controlled per M7. The acceptability of key analytical procedures for their intended regulatory purpose is critical, and as such, pertinent method validation should be provided per USP and ICH recommendations. Reference standards used should be adequately qualified, and for USP reference standards, lot number and COA are sufficient. However, for other standards, data for structural elucidation and characterization should be provided. Some common deficiencies for drug substance are related to solubility. Knowledge of BCS solubility is critical in the establishment of risk ranking for critical attributes based on the potential impact of drug product performance, manufacturability, and stability across dosage forms. For example, in the case of liquid filled capsules or really any liquid formulation, solubility characteristics may increase the risk of crystallization during manufacture or during stability, during shelf life. Therefore, it's critical to understand the solubility attributes of your drug substance. So regarding polymorphs, it is critical to demonstrate knowledge of drug substance polymorphism by, referencing the current guidance for industry on polymorphs, and in particular when polymorphs may have differences in stability and solubility. Force degradation studies are important as these not only help confirm the stability indicating properties of the methods, but also provide critical information on the main degradation pathways. Therefore, attaining adequate degradation and reporting known and unknown impurities are important aspects of these studies. So regarding impurity structures, it is important to ensure high resolution and accurate structures of impurities to help support our efforts with KASA. Next, I will discuss drug product assessment, where I will cover a few aspects of formulation, drug product controls, release and stability, the container closure, and stability. Okay, so formulation, in terms of composition, know that in general, drug substance overages are not a recommended practice. If overages are required, these should be well documented and justified. And then for excipients, excipient selection should be justified based on a well-designed studies that include a consideration of the ratio of excipient to API and the chemical reactivity of excipients or potential impurities in excipients. These studies provide one piece of data of potential incompatibilities and should be used together with data obtained during manufacturing development and stability studies to obtain a complete picture. Likewise, data to support the grade and level of excipients and the use of stabilization agents in the formulation. It's critical to be, so it should be included in the submission, and compendial excipients should be controlled based on compendial requirements. As usual, additional material attributes can be established based on risk. For drug product controls, for release and stability, similar to the drug substance controls, compendial drug products should comply with the current USP monograph, and inclusion or absence of additional control strategies should be justified based on a risk-based approach. The assessment of risk should be supported by data in the product development report. For example, if your API has multiple polymorphs and polymorph testing is not proposed, then the PDR should include data to justify absence of testing. Upon initial submission for solid oral drug products, the in vitro drug release method and acceptance criteria will be evaluated by Biopharm, and revisions may be recommended. So a revised dissolution acceptance criteria should be justified based on the drug product profile. So in particular for modified release dosage forms, the reporting of full drug release profile results during stability may help prevent the need for a new batch to show compliance of the release profile at initial time points, as well as to support the proposed shelf life. So, and then as discussed for the drug substance, analytical methods should be demonstrated as suitable for the intended regulatory purpose through adequate method verification and validation per current USP and ICH guidance. So I'm going to discuss a little bit more in terms of drug product controls for impurities. So control for impurities should be based on current guidance, and in general, substances identified as process impurities in the finished API may not need to be controlled in the finished drug product, but know that on occasion, an impurity that is identified in the API as a process impurity by the DMF holder may be a potential degradant in the finished drug product as it undergoes processing. So knowledge acquired during product development and impurity profiling can help address these issues up front to avoid extensive work during ANDA assessment. In addition to the recommendations per ICHQ 3B, we also recommend following ICHM7 on the evaluation of the genotoxic potential of impurities. So, and as previously mentioned, whenever an impurity exceeds the recommended limits, either references from the literature and or PharmTox studies should be provided as justification. Residual solvents reports and the elemental impurities risk assessment should be provided to show compliance with USP and ICH requirements respectively. And then in terms of nitrosamine impurities, these should be evaluated based on the current guidance document. And this topic, this is a topic that is continuously evolving. So I invite you to not miss the presentation later on in the workshop. The container enclosure selected should provide adequate protection over the proposed shelf life and should be shown to comply with USP 661 and 671. The data package should include certificates of compliance with applicable 21 CFR requirements for indirect food additives, and keep in mind that for combination drug products, a consult may be required. So in terms of stability, the initial submission should include 6-month accelerated and long-term data for all 3 exhibit batches manufactured using at least 2 lots of drug substance, and bulk packs intended for repackagers should also be added to the stability program. In-use stability data should be generated for drug products intended for alternative administration, for example, tablets dispersed in liquids, or whenever the capsule contents are intended to be sprinkled on soft foods. And in addition, the need for in-use stability may be determined based on a risk-based approach. For example, a drug substance with significant sensitivities to environmental conditions may require in-use studies to establish acceptability. Additional stability data should be submitted to justify the expiration date per ICH-Q1E prior to approval. And expiration date extension should be described in the application, keeping in mind that for products approved with a reduced expiry due to failures on accelerated conditions, any extension of the shelf life should be requested in a supplement to the agency. And I also refer you to the ANDA stability testing of drug substances and drug products guidance for industry and question and answers as a really good source of information for questions on stability. Some common issues during drug product assessment result from inadequate compatibility studies. As mentioned, it is important to consider the ratio of excipient to API and to test and report observed impurities. One of the most common issues involves water content limit that is not supported by data. In order to ensure products of consistent quality, the control strategy and limits should be based on data in the submission. In terms of data trends, failures, and lack of mass balance of ASEAN impurities, these are critical, in particular for those instances in which a shelf life is extrapolated based on ICH-Q1E. So if you see these, please discuss them and justify them through demonstrated product knowledge, and if necessary, provide additional supporting documentation. These will definitely save a lot of review time. As mentioned, lack of dissolution data to support the agency's recommended acceptance criteria can result in a significant delay if a new batch is needed. So sometimes the best way to avoid any unnecessary delays is to provide dissolution profiles for your drug product during stability. Failure to use a risk-based approach to establish the need for in-use stability results in delays due to the need of additional stability studies. Regarding the stability protocol, whenever a scale-up is proposed, validation lots should be added to the stability program under long-term and accelerated stability. And note on requests for final approval of tentatively approved applications. These requests should be accompanied by an outline with updates that include location of changes and justifications as needed. Furthermore, since at times the approval requests come a significant number of years following the tentatively approved action, please confirm compliance with current regulatory requirements. For example, if a USP monograph became official during that time, then all supporting documentation to show the drug substance or drug product comply with current requirements should be provided. Not doing so can result in significant delays due to the data required prior to granting full approval. In this case study, the drug product uses an API that is a mixture of polymorphs, in this case, say 1 and 2, each with distinct solubilities, so the ratio is critical for acceptable bioequivalence. Polymorph 1 is stable, and 2 exists at a specific water content, and it is known to interconvert to other forms. Formulation development studies show specific polymorph ratios fail to achieve bioequivalence. So with the knowledge acquired during product development, the applicant was able to establish adequate controls and limit water content to prevent interconversion of Form 2, and establish a control to monitor the polymorph ratio in the finished drug product with a limit that was supported by data. So this is an example of how knowledge gained during product development was used to establish meaningful control strategies to ensure a product of consistent quality is manufactured. This is my challenge question for you. Which statement is not true? A, pharmaceutical quality assures every dose is safe and effective, free of contamination and defects. B, OPQ quality assessment is a multidisciplinary task and employs science and risk-based policies and standards. C, Product development studies to generate the needed product knowledge and understanding are critical for a high-quality submission. And D, data trends on stability do not need to be evaluated. So take a couple seconds to try to come up with the correct answer. And if you selected D, data trends on stability do not need to be evaluated, you are correct. This is a very important aspect, actually, of the stability discussion and should always be pointed out, and any additional supporting documentation should always be provided. In summary, I've shown you how industry and the regulatory agency must work together to ensure high-quality drug products. I've described the IQA as a multidisciplinary approach to ANDA assessment and the functions of the drug product discipline within the IQA. I've also identified some of the most common deficiencies encountered by the drug product discipline and provided some strategies to reduce the presence of these in the ANDA submissions. If you are to take one message from my presentation, let that be that demonstrating product knowledge and understanding is probably the most effective tool for a high-quality submission, and a high-quality submission can result in a shorter time to approval. So it's a win-win for everyone. With that, I conclude my presentation. I will be back to answer any questions you may have during the panel discussions. Thank you for your attention.
We'd like to thank all of our speakers for the excellent presentations. And as we move into our question and panel discussion, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks as if we have some questions coming in right now. The first group of questions will be for Peter Capella. And here is the first question. Is this system that's used by FDA only, or going forward, does FDA want generic manufacturers to use this system?
Good morning. Yeah, this is Peter. It's a very good question, and actually there were several others that came in with a very similar point of discussion. And I did want to just start with saying this is really an internal FDA system for use by assessors, whether they're being drug product, processing, or biopharmaceutics. So we're really showing this to you and to everyone as sort of in the spirit of transparency as how we do our thinking and our most recent version of that assessment. So we really don't request generic manufacturers to make any changes to their submissions based on KASA. And another question related to that was about ECTD formats. People are like, well, KASA doesn't appear to be exactly like in ECTD. Do we need to think about changing it? And my quick answer to that would be no. There's really no plans based on KASA to make changes to the well-accepted ECTD format. And in fact, KASA was designed to, and our assessors are very familiar with ECTD, so we're designed to assess ECTD formatted applications using the current version of KASA. And if ECTD does, modify in the future, we'll make modifications appropriately to the internal KASA structure as well. So hopefully I answered that question. Thank you.
Thank you for responding to that question. We have another question for Dr. Capella, and here is the question. So does the FDA reviewer have to enter all of this information into the KASA manually? And is there any talk about the applicant entering this data in the future?
Yeah, it's another great question. And believe me, our assessors, as we were developing KASA, had similar discussions. And effectively, we're trying to strike a balance between the information that is obtained and collected within the assessment. So the quick answer is some data is auto-populated from submission data, but some is not at this point. It's kind of a mixed bag in that case, but we're trying to, again, structure the data in a way that it's easily mineable or comparable. But at this point, you know, as I said earlier, we're able to take some data directly from the application. And in the future, I'm sure that can be improved and updated. Of course, from our perspective, you know, the data is critical, but the most critical thing is the evaluation of the data, of course, and also assessment of risk. At this point, you know, we do require some input of information by our assessors. The good news is that once it's into the assessment, it can be retained for future updates to that assessment. So it's basically like a living document. So as the assessment process matures, leading to ultimately to approval, we hope, that same information can be updated with responses to questions, et cetera. So thank you.
Thank you for responding to that question. We have one more question for Dr. Campello in this round, and here's the question. How far back does the ANDA reviewer go to use data analytics if the drug substance drug product is already approved, and if it is a first generic other than the NDA, what analytics are there? And bear in mind, at times the NDA data may not be transferable to the ANDA.
Thank you again. This is, again, a very good question, and it points to a very interesting case. Of course, the first generic, often cases there are multiple first generics based on the same day of application to a given NDA. But I agree with the question that obviously, really all you have for your pharmaceutical equivalents for the first generic is the NDA itself. And again, here as FDA, we strike a balance. We realize that all the information that we receive, whether from an ANDA or from the NDA, are confidential and certainly proprietary to the company. So our goal is to respect that confidentiality, and also that, as I mentioned in my slides, even at prior to KASA and as part of our current IQA process that Myra outlined effectively, every application has to stand on its own merits. So that goes without saying almost, because again, we have regulation and guidance as our guides to make the assessment. So the benefit of having this ability to look at different applications is mainly for comparative analysis, because again, many cases there's very black and white requirements, but in certain cases there are gray areas, and those, especially in those cases, having the data to compare across applications helps improve our consistency with our decision making, as I mentioned in my talk, from the entire life cycle. So from the original acceptance of the NDA itself through the first generics, which again, data is somewhat limited, but then ultimately that the goal is down the road as generics, as the life cycle matures, we'll have more information at our fingertips, especially for post-marketing folks who need to make decisions on supplemental changes fairly quickly, and they will now have the ability to look at this data to help in those decisions. Thank you.
Thank you, Dr. Capella. Our next group of questions is directed to Rocky Shah. And here's the first question for Dr. Shah. Does the ANDA applicant need to list the facilities used for excipient and packaging component testing on the 356H and in 32P30?
Thank you for that question, Ray. So no, the applicants are not supposed to list their facilities which are used for excipient manufacturing, testing, or packaging component testing. The 356H guidance and the question answers that we have published clearly states that we need the facilities which are used for commercial manufacturing of drug substance, drug product, testing, as well as any critical intermediate facilities. Those need to be listed. So these facilities do not need to be listed in 356H form.
Thank you for responding to that question. We do have another question for Dr. Shah, and this is the question. Does the KASA also apply to NDA manufacturing assessment?
Definitely, yes, it will apply, but currently we have started with generic applications and we have started with only solid oral to begin with. We cannot get everything at a time because this is a kind of a paradigm shift, if you could see from Peter, Mai, and Kimberly's presentation. So it takes time. So NDAs are in the pipeline. The first thing was to get very high volume, which is ANDA generic solid orals. It will be followed up with drug substance now. That will be coming up later this year. After that, we are going to get liquid applications into KASA, and this will be followed up by NDAs. So NDAs is in the pipeline, and it's not too far. Thank you. I would also cover some of the questions that were on different dosage forms. So right now, as I mentioned, we covered only solid oral in this presentation because that is live. But going forward, we are getting all other kinds of dosage forms. So liquids, semi-solids, and some of the complex dosage forms will follow in KASA. So yes, all the dosage forms will be done in KASA and all different type of application. And it's not only NDA, but even BLAs are planned for KASA. So BLAs and followed up by supplements. So in future, we will be doing most of our assessment in KASA. I hope I answered majority of questions on what type of applications and what kind of dosage forms.
Thank you for responding to that question. One more question for Dr. Shah in this round. Is the industry content of the manufacturing risk assessment and control module available to the
So when it says the content of the module, so we have our risk assessment algorithm that we have developed internally. So they are not available to industry, but again, it's nothing like rocket science. You could see from my presentation that we look at API information, excipient information, as well as formulation information, in addition to any scale up or control strategies that the applicants could be using. And we have included even facility type of factors. For example, what kind of experience the facility has in manufacturing those kind of dosage forms or unit operations. We are going down to the unit operation level through KASA, and also what kind of control strategies were already in place and what kind of data issues we have found from the facility. So all that is combined, and we have built an algorithm which is more like a quantitative algorithm. We have used failure mode and effect analysis, but that is not available at this time because again, this is an internal risk assessment process and it's just initial risk assessment. And based on what we find from the applications and by the say information request route, we can adjust the scoring that comes out, and we do our final assessment by looking at the risk mitigation strategies. So it is still kind of in the pilot stage, if I could say, that we are trying out and if we are trying to make the model better, and it will be helpful in assessing our application risk assessment for our applications. Assessors or reviewers can modify the score right now. So we know that in 100% of time it may not work, right? Because we have now on our in-depth review of the application, we find that the applicant has done this kind of risk mitigation that was not apparent when we do the initial risk assessment. So that's why it is not available right now. But again, majority of the drop downs are built upon what we find from reviews and what mitigation strategies we have seen across applications in doing so many years of reviews for these applications. So it is built on that. So and a lot of times it is available in all other types of presentation, but it is just this module is not available right now to industry. Thank you.
Thank you for responding to those questions. We're moving on to our next panelist, Kimberly Rains. And here's the first question for Dr. Rains. Why does the agency now require for IR products a dissolution spec of 80% Q in 15 minutes, even though the existing USP monographs have specific dissolution?
Thank you for the question. So all of our dissolution specs are drug product specific. We do recognize the USP dissolution methods and tolerances that are set, but we would evaluate the data on a case-by-case basis. If a drug product meets the standards of our current guidance for highly soluble drug substances, we do prefer the standardized dissolution method, which has acceptance criteria of not less than 80 in 30 minutes. However, if alternate methods are used, we would set an acceptance criteria that is appropriate. Thank you for the question.
Thank you for responding to that question, Dr. Rains. We do have another question for you, and here's the question. For high and very high risk DP product that has demonstrated BE, what else is the agency looking for regarding dissolution?
This is a good question because our understanding is if they're able to demonstrate bioequivalence, it should mitigate the risks that we would see in any perceived differences in vitro dissolution. Our expectation is that the in vitro dissolution is predictive or somehow influenced by the in vivo performance. So we would want to see that the in vitro dissolution tests could predict or be related in such manner. This could be through an IVIVR or with additional data or understanding of the knowledge of the drug substance and drug product to inform our decision.
Thank you for responding to that question. We're going to move on to our next panelist. We've only got a few minutes left. And this is for Mayra Pinheiro-Sanchez. And here's the first question. If the applicant is aiming to comply with recently used DS and DP USP monographs by filing the USP methods as an alternate test methods, USP and in-house methods are equivalent and impurity profiles are compatible, but to retain the higher, tighter in-house specifications, can a USP claim still be made on the label?
Yeah, good morning. Yes, the USP represents the minimum requirements, so implementing tighter impurity limits, for example, will ensure the drug product meets the compendial status. So the answer to your question is yes.
Thank you for responding to that question. The next question for Dr. Sanchez. Is it required to perform excipient compatibility study if I use excipients the same as the RLD in my generic application?
Yeah, the use of the same excipients in the RLD can be used to further mitigate risk. But for example, for solid oral drugs, the ratio of excipient to API may be different. In addition, on occasion, impurities in excipient might have an impact on the stability of the API. And so this knowledge is, also important to address any potential problems that may arise during the commercial stage. So yes, I mean, excipient compatibility studies should be included in the application.
Thank you, Dr. Sanchez. We're going to try to squeeze in one more question in our Q&A, the first Q&A panel. And here's the final question for Dr. Sanchez. Do I need to report the process impurities?
Yeah, so obviously for drug substances, that's a yes. All impurities, process, and degradants need to be reported. With respect to the drug product controls, process impurities are not required to be monitored, as they're not expected to increase over time, and they're not supposed to be affected by the manufacturing process. But keep in mind that at times the impurities that are identified by the DMF as process impurities may be found to be degradants in the drug product. So it's important to be aware of the impurities during method development and validation. And if you see during those studies that an impurity that's identified as a process impurity increases, then we ask that it be noted and that that impurity be monitored as a degradant in the drug product. So it's important to have an open mind in terms of the degradation characteristics of the product and, what impurities could be the result of degradation. Thank you.
Well, that's all the time we have for questions in our first Q&A panel. And we want to give a huge thank you to all of our panelists for answering numerous questions that came in. We'll now enter into